Good morning, everyone. We will start in 5 minutes. Good morning, everyone. Welcome to Global and Local Perspectives on the Regulatory Frameworks for New Breeding Techniques in Crops. This webinar is part of a series organized by ISA Inc. in collaboration with the DA Biotech Program Office. I am Janine Escasura from ISA, and I will serve as the moderator for today's event. New breeding techniques have transformed agriculture through the precise, efficient, and cost-effective development of high-yielding, disease-resistant, and climate-resilient crops. The advancement of these technologies calls for the need for stakeholders to stay informed about the latest developments in biosafety regulations. The first of the series will provide an overview and updates on the regulatory pathways related to crops developed through NPTs. As of now, we have more than 100 participants joining us today. Please be reminded that this webinar will run for 2 hours and will be recorded for documentation and evaluation purposes. Before we start the webinar, please allow me to quickly remind you of the house rules. First, Zoom attendees are requested to keep their microphones on mute unless invited to speak. We highly encourage our audience to engage and actively participate during the Q&A session. You may post your questions using the Zoom Q&A button seen at the bottom of your screens. Please do not forget to indicate to whom you are directing your question. These questions will be addressed during the Q&A session. Lastly, please complete and fill out the online evaluation form to receive your certificate. A link and QR code will be flashed at the end of the webinar, and only those who have accomplished the post-webinar survey will be given the ACE certificates. We also have a set of activity objectives that we would like to achieve at the end of the webinar. First, this webinar intends to provide information on the biosafety regulations and policy frameworks governing the use of NBTs in crops. This webinar also aims to expound on the challenges and potential policy directions that can support the responsible use and sustainable use of these technologies. Lastly, this webinar intends to be a venue for stakeholders to engage in meaningful discussions on harmonized regulations, facilitating the responsible use of NBTs in agriculture. To formally start our webinar, may we call on the Executive Director of ISA Inc., Dr. Rodora Romero Aldemita. Dr. Aldemita is a multi-awarded scientist and science communicator. She holds a PhD in botany from Purdue University and a postdoctoral fellowship on golden rice at Albert Ludwigs University, Germany. She coordinates the activities of ISA's Global Knowledge Center on Biotechnology, as well as capacity building activities on biotechnology and biosafety in Asia, in partnership with USDA and the Philippine DA Biotech Program Office. She is also a member of the technical advisory team of the Applied Biotechnology Committee of DABPO, a board member of the Biotechnology Coalition of the Philippines, the editor-in-chief of the Philippine Journal of Crop Science, and the scientific reviewer of 6 scientific journals. Everyone, please welcome Dr. Rodora Romero Aldemita.
Hello and good morning, good day, good evening, everyone. Welcome to the ISA webinar series titled Global and Local Perspectives on the Regulatory Frameworks for New Breeding Techniques in Crops. So this is the title of the webinar today, which is the first of the series of webinars on regulation. We are the International Service for the Acquisition of Agri-Biotech Applications, a nonprofit organization based at the International Rice Research Institute, Los Baños, Laguna. We envision a world where accurate biotech information empowers farmers and consumers to drive food security through innovation. So our mission is to feed the world with knowledge and biotechnology by supporting transformative technologies, fostering impactful partnerships, and facilitating the development of science-based policies towards food-secure communities. So this webinar, as I said, is part of a series, and our topics today will be provided to us or discussed to us by 2 noted experts. And this is about new and emerging biotechnologies in the global and the Philippine context. So after the webinars on gene editing, gene drives, and novel foods that we had in the past few months, and also at the end of last year, we are now looking at the regulatory frameworks. So the first one again would be on regulatory framework or regulatory considerations for new breeding technologies. And we are very pleased today that we have participants from many regulatory agencies, not only in the Philippines, but also outside the Philippines, regulatory system experts, policy experts, researchers, partner institutions, farmers, farmer groups, and of course, the general public. And everyone is interested to learn these topics. So once again, bioscience innovations such as gene editing transform agriculture through the precise, efficient, and cost-effective development of improved genetics, not only in plants, but also in animals. We already learned that there are plants and animals which are improved plants and animals derived from gene editing that are commercialized, and the Philippines has already provided a positive consideration for some of these crops. So in today's webinar, as I said, we will feature two experts. One will talk on the Philippines, and another expert will talk about the global perspective and how is the regulatory trend, what's happening, what are the discussions going on, what are the um, foresights and hindsights on all of this. They will also discuss the challenges and potential policy directions that can support responsible and sustainable use of these technologies. This webinar will thus serve as the venue for stakeholders to engage in meaningful discussion, and we would like to hear from everyone through your Q&A questions. So this is really a venue so that we can have an exchange. One from everyone. We have our experts from the Philippines and one from Australia. And so we hope that through these discussions, we can get ideas and possibly pointers to harmonize regulation so that we can facilitate adoption and acceptance of products of new breeding technologies in food and agriculture. So we hope that by the end of this 2-hour webinar, our esteemed participants, especially the regulators and policymakers, I hope you will stay on and you will have understood and appreciated the featured NBT framework to ease product determination. And our young and aspiring scientists, I see some names already greeting good mornings and greeting everyone. I hope that when we finish, you would have been inspired to get to your biotechnology career track. And of course, the general public, I know that there are some in the audience, and I hope that you will appreciate what we have to offer today. We also invite you to visit our ISA website. We have many publication materials and articles and teaching materials so that you can help your colleagues learn about new breeding technologies and the regulations that govern it. So with that, happy learning, everyone.
Thank you very much, Ma'am Ola. Now, please allow me to introduce to you our first speaker for today. Ms. Maria Lorelai Yu Agbagala is an assistant scientist at the Department of Science and Technology. She is also the head secretariat of the National Committee on Biosafety of the Philippines and the DOST Biosafety Committee. As the secretariat to both committees, she leads the secretariat in providing administrative and technical support to both committees in all biosafety and biotechnology-related activities. She also leads in monitoring and the conduct of horizon scanning on global developments in modern biotechnology, biosafety regulatory issues and concerns, and new and emerging techniques in support of the policy formulation function of NCBP. Ms. Agbagala is also a member of the technical working groups involved in the crafting and finalization of the revised Joint Department Circular Number 1 Series of 2021, formulation of regulatory framework on genetically modified animals, and development of guidance document on genetically modified fish. Ms. Agbagala was awarded the Fulbright Philippine Agriculture Advanced Research Grant in the USA at the Fort Lauderdale Research and Education Center in Florida. From 2016 to 2018, she was the focal point for the FAO GM Foods platform. To discuss regulating innovations, new breeding techniques in the Philippines, please welcome Ms. Maria Lorelai Yu Agbagala.
Yes. Good morning. Thank you, Miss Janine, for the kind introduction. Okay, so let me first greet everyone. A good morning, good afternoon, good evening, wherever you are. And it's really a pleasure to be here with all of you today. So are we ready with my presentation?
Yes, Miss Malu, can you see your presentation?
Okay, is this the one? Okay.
Yes.
Yeah, I actually requested Miss Tin to run my presentation. So okay, so for today, I'll be sharing insights on regulating innovations, new breeding techniques in the Philippines, and this presentation will describe how the Philippines is adapting its regulatory system to keep pace with the advancements in biotechnology. So we'll explore current biosafety framework, key case studies, and the future policy direction for NBTs in the Philippines. And I think with so much happening in the world of biotechnology, it is crucial that we strike a balance between innovation and safety. Okay, so this presentation will run like what you've seen on your screen. First, we'll start with defining new breeding techniques, what they are, and how they differ from conventional breeding methods and genetic modification. The next, we'll take a look at the Philippines regulatory system for NBTs, how we currently assess and manage these technologies within our biosafety framework. Then, to actually to have a concrete example for this technology, we have real world example like the Tropic Biosciences Reduced Browning Banana and Sanatec Seeds GABA Tomato. And then finally, we'll wrap up with the presentation on policy directions for NBTs, where we are, ongoing conversations, and how we can ensure that our biosafety regulation or our regulatory system remains science-based, risk proportionate, and supportive of innovation. Next slide. Okay, so before we get into the specifics, let's first define new breeding techniques or plant breeding innovations. I'm sure you will agree with me that these techniques or this new these plant breeding innovations are really a game changer in crop development. And these are advanced molecular, genomics, and cellular tools that help us create new plant variety with specific desirable traits faster and more precise than conventional breeding methods. And these techniques actually allow our scientists to make targeted changes in the genetic level, making the process more efficient and predictable. So instead of waiting years for these traditional breeding methods to achieve the desired traits, these techniques really are make the process easier and more efficient and faster. Next slide. So again, with these new breeding techniques, we can make precise changes to an organism's DNA, and depending on the technique used, we can introduce we can introduce a simple change in the genes, or we can delete a small DNA segments, or insert a few bases. And what's interesting is that the resulting organism could have occurred through spontaneous mutation or the traditional physical and chemical mutagenesis. But with NBTs, we can get there in a more targeted and precise way. Next slide. Okay, so to in a nutshell, and just a brief review of our Biotech 101, this conventional breeding method uses traditional methods like cross selection and breeding to develop a new plant variety. And this method is actually less precise because it introduces many genetic changes at the same time. So it requires multiple generation to refine the trait. While genetic engineering or genetic modification, it actually involves insertion of foreign DNA from an unrelated species to introduce a novel trait into the plant's genome, like you have For example, we have our herbicide-tolerant corn or insect-resistant corn. And genetic engineering is actually strictly regulated because there are concerns on environmental and food safety. While for the plant breeding techniques, well, it actually uses precise gene editing methods like CRISPR and TALENs to make targeted changes in the organism's DNA. And this is actually more faster and efficient than conventional breeding method, while being more targeted than genetic modification. Next. Okay, just a brief background on how we come up or how we develop our policy on new breeding techniques. In back in 2018, the Department of Agriculture commissioned or put together a study group to explore the regulatory landscape of these new breeding techniques. And their findings were then presented to the National Committee on Biosafety, which led to a recommendation for our country to develop or establish our policy on these technologies. So by 2020, the NCBP Resolution Number 1 was issued, marking a key step in setting up the regulatory direction for NBTs in the Philippines. Then in 2022, the Department of Agriculture issued a Memorandum Circular Number 8, which laid out the procedure for the determination of the products of these plant breeding innovations. And in 2023, we saw a major milestone. The Philippines made its first ever PBI product determination. So we have this gene-edited banana with reduced browning, which is actually officially classified as a non-GM, demonstrating that our country is really moving forward in regulating these technologies. Next slide. Our policy on new breeding techniques basically sets the rules for classification or for classifying the products of these new breeding techniques. And it's actually really very simple. If your product contains a new combination or novel combination of genetic material introduced through modern biotechnology, then it's classified as a GM. Otherwise, if otherwise, the product will be considered a non-GM. And the policy also states that only PBI-derived GM plants will be subject to regulation under our existing framework for GM. And it also tasks the Department of Agriculture to develop the guidelines for the evaluation and monitoring of these products of PBIs. Next slide, please. So after the issuance of the policy on NBT in 2020, the rules and procedures for the determination of products of PBI was issued in 2022. And this is actually very important because as these new breeding techniques continue to advance, we need a clear science-based approach to assess these products and to determine whether they should be regulated under our GM regulatory framework. So this guidelines actually provides a systematic way to assess whether this plant breeding innovation will result in a product that will qualify as genetically modified, or will it fall as outside that definition. Next slide. So this is the first step in the process, in the determination process. We have a technical evaluation to determine whether the final product contains a novel combination of genetic material. And then the BPI actually put together a group of experts to do the evaluation and the determination. And I think two of the members should come from BPI, and there would be one external expert if they deemed necessary. And who can submit a request for determination? So a product developer, and it could be a department, any department or agency in the Philippines, a university with research institutions in the Philippines, international research organizations, corporation registered with the Securities and Exchange Commission, and any cooperative registered with the Cooperative Development Authority. And if you're a non-resident, you shall appoint an agent who is a resident of the Philippines before you can be able to have your request evaluated. Next slide. Okay, so let's take a closer look at the timeline of processing of these submissions. First, we have this pre-assessment, which takes about 3 working days, and this is where the initial review was conducted, and this is to ensure that all the documents and all the requirements are complete. Then we have the conduct of the evaluation, which takes place over 24 working days. And during this period, the submission will undergo a technical review by the expert. And basically, one of the major requirement is for the product developer to submit experimental evidences that demonstrate that there's no new genetic combination in the final product. And at the same time, there is a public commenting period over 10 working days. There is this public information sheet that contains all the relevant information about the product. And this will be posted at the BPI website. And this actually allows our stakeholders to provide feedback and raise their concerns over the product. And after this evaluation and this commenting period, there would be an official determination. An official determination will be issued within 5 working days, and this actually is the time where the official determination or the final decision on the regulatory status of the product will be officially released. So overall, the whole process is designed to be efficient, transparent, and science based, while ensuring there is proper regulatory oversight. Next slide, please. So what is this? What is this official determination process? So once the expert group, the BPI expert group, completes its evaluation and the public comments were considered, the director of BPI shall make the official determination or shall make the final decision on the regulatory status of the PBI product. And this is actually basically based on the recommendations made by the TISED group. And there will be two scenarios under this process. Number one is if the product is determined as a GM, then the BPI will inform the product developer that their GM product will be subject to regulation under our existing regulation, which is the Joint Department Circular, and further advise the product developer to proceed with their application under this regulatory process. And since this is classified as a GM, then the BPI will also monitor this particular product for compliance with our regulation. And then another scenario is if the product is determined as an non-GM, then the BPI will issue a certificate of non-coverage to the product developer, and this will be posted at the physical website at the BPI website to inform the public of this decision. Next slide, please. Certificate of non-coverage, what it really means? Well, this certificate is specific to the novel characteristics that is introduced into the variety, and it also applies to all its future generations, meaning all related plants that inherit the same trait. Then the certificate also apply to all genetic background that includes this characteristic, whether they are developed by the product developer or through their licensees by further breeding. But take note that this certificate of non-coverage does not excuse or does not exempt the product developer from complying with other regulations set by the Department of Agriculture and also by other government agencies. So it is very clear that this certificate of non-coverage only exempt the product from being regulated as GM, but the product developer will still have to comply with with all other applicable laws under our Philippine regulatory system. So next slide. Okay, so this is an example of a certificate of non-coverage, and you can see that there's detailed information on the product and also on the product developer, and there is that official determination on the regulatory status of the PBI product. Next slide. Next slide. Next slide. Oh, what happened? Next slide. Okay. And okay, so to continue, okay, you'll also see a statement of compliance, which makes sure that all regulations or all rules and procedures have been considered. And then there's a validity period to make it clear how long will the certificate remains effective, and also a date of issuance, which is when was the determination made by the BPI director. Next slide, please. Another click. Okay, so this is one example of the determination that was made by BPI for high GABA tomato, and it was you would see that this product was also determined as a non-GM in Japan, and also it is there regulated by the U.S. Department of Agriculture. Next slide, please. Another click. So, looking at the global regulatory landscape for gene-edited crops, I think it is safe to say that that the Philippines approach aligns with the regulatory trends in other countries or in other regions. While different economies or different countries have slight different criteria on NBTs, the trend is shifting towards a risk-based product focused approach, which is similar to the Philippines. And in fact, the European Union is actually have ongoing discussions to update their position or their treatment on these products of new breeding techniques. Next slide, please. Another click. Okay, this slide actually captures the key elements needed for a science-based determination when it comes to regulatory decision making. And looking at this slide, I would like to think that the Philippines really is in the right direction. Why? Because, like for example, it says that there's a need for a clear definition and criteria. Well, our resolution or our NBT policy actually has well-defined terms that determine whether a product of NBT will be regulated or not, and which product will be subject to our regulatory oversight. So next is, do we have an applicable policy or regulation? Yes, we have this Department of Agriculture Memorandum Circular Number 8, which provides the legal framework for decision making. How about implementing regulatory authority? Yes, we have the Bureau of Plant Industry that implements and enforces these policies. And for stakeholder awareness and engagement, I know this is very, very critical and this is very essential. That's why because we know that they have to make sure that that the community, the public, our scientists, our regulators understand and trust the process. So there have been continuous public consultation, stakeholder consultation in order to make the public aware of this technology. And to support this activity, capacity building and expertise development is the key. So our regulators are continuously participating on trainings and workshops to equip themselves with the right knowledge and tools for this particular technology. And last is international collaboration. We are actually we acknowledge, we are aware on the importance of international collaboration or collaborating with other with our partners, because it helps us it helps us to learn from global best practices and to harmonize our approaches across countries. So together, these elements strengthened the credibility and effectiveness of our science-based regulatory system. Next slide, please. So these are just the examples of products that have undergone determination by the Bureau of Plant Industry. We have like three reduced browning banana and one determination for this high GABA tomato from Sanotec. Next slide, please. Okay, so this slide will show that this new breeding techniques is already driving innovation in our agriculture sector. Like for example, at Erie, genome editing or gene editing is being used to develop rice varieties for sustainable farming to help increase yield, resilience, and efficiency. Meanwhile, at PhilRice, the DA Crop Biotech Center also has ongoing projects also on rice improvement. While over at UPLB, at the Institute of Plant Breeding, there are ongoing activities to develop varieties using these cutting edge technologies. So next slide, please. Another click. Okay, so the next question is what's next for new breeding techniques in the Philippines? So it's actually one of the main point for this presentation. So next slide, please. Well, I think I believe that our NBT regulation is aligned with most of the countries, ensuring consistency on how NBTs are assessed. And our key research institutions such as ERI, PhilRice, UPLB, and PICARD are already exploring or working on these applications for our some of our Philippine crops. However, to make this NBT successfully adapted for successful adoption of these NBTs, then these building programs are really crucial. So we really need to equip our regulators with the right knowledge and tools to address the concerns or to be of knowledge or to be more knowledgeable on these technologies. And lastly, it's just as important that we educate our stakeholders, our farmers, and the public on NBTs, what are they, how they work, and their potential benefits. So next slide, please. Another click. And so another question is, are we considering developing a policy on gene-edited animals? Well, recently we have been receiving queries from companies abroad that are interested of bringing in these products of gene editing. And just a few weeks ago, we received a letter from the Department of Agriculture, the Bureau of Animal Industry, actually requesting the committee, the NCBP, to probably look into issuing or having another policy for this for gene editing in animals. And well, the actually right now, like next slide, please. Another click. So in response to that request from BAI, well, the BAI, Bureau of Animal Industry, is already preparing the groundwork for gene editing in animals. Actually, we have had several meetings, several discussions, and one that when it was like 2 weeks ago or 3 weeks ago that we had a meeting with the product developer. This is actually via Zoom, and the officials of the DA were also present, and it's actually really encouraging because the DA is really facilitating or tracking the process and really giving their support in order for this undertaking to have to be under this undertaking to be um, to move forward. So another next slide, another click, and we already have drafted the guidance document for GM and gene edited fish. And so these are just some of the things that are undergoing right now. And we know that this is not easy. We don't want to raise our hopes too high. Uh, this is not a walk in the park. We know that there are still some steps that we have to go through, like regulatory reviews, stakeholder consultations, documentation, but things are definitely moving in that direction. So next slide, please. So I think this is my last slide. For key message, well, the Philippines is making sure that as we adopt these new breeding techniques or plant breeding innovations, we do it responsibly. We have a regulatory system that is science based and can adapt as the technology evolves. And our objective is to ensure that these innovations are safe, sustainable, and most importantly, to earn the trust of the public. So it is really about finding a right balance um, between supporting innovation while ensuring safety and accountability. Next slide, please. Okay, so let me just thank the following people for providing the materials for this presentation. I'd like to thank Ms. Emma Eusebio and the BPI Biotechnology Office, Dr. Ordonio, Reynante Ordonio of PhilRice, Mr. Raymond Corona, and the ISA for the infographics. Next slide. And thank you. Thank you for listening. Thank you.
Thank you very much, Ms. Malu, for giving us an overview of the policies and regulations governing the use of new breeding techniques in the Philippines. Everyone, you may send your questions through the Zoom Q&A function, and we will answer them later during the Q&A session. Again, thank you very much, Ms. Malu, for that very comprehensive presentation. Everyone, let us give her a virtual round of applause. Moving on to our next speaker, we have Dr. Michael Jones, a professor from Murdoch University. Dr. Michael Jones has a degree in biochemistry and PhD in plant biochemistry from Cambridge University, UK. After postdoctoral positions at the University of Missouri, the Australian National University, and Cambridge University, he held positions at the Welsh Plant Breeding Station and then at the Rothamsted Research, UK. He then became a professor of plant sciences at Murdoch University in Western Australia and a foundation director of the WA State Agricultural Biotechnology Center in 1993. He has a wide range of experience in R&D on crops, having worked in departments of biochemistry, plant pathology, developmental biology, biological sciences, biotechnology, and plant breeding. He is committed to the translation of research outcomes to commercial implementation. His work has included the molecular basis of nematode plants interactions, plant virology, molecular markers for crop improvement and transgenic technologies, and pioneered biotech-based agricultural R&D in WA. He has served as on Australian Commonwealth and state government committees, generating more than 320 scientific publications, supervised over 70 PhD students, and co-founded 2 companies. Current work is on new breeding technologies, gene editing for crop improvement, and the international regulation of gene-edited crop produce to promote trade. To discuss a global perspective on regulating crops developed through new breeding technologies, please welcome Dr. Michael Jones.
very much. Oops, what am I? Oops. Share presentation. Sorry, this was all worked perfectly before. Right. Share. I'll still move down a bit. Here we are. Share. Okay, can you see that?
Yes, we can see your screen now.
Okay, thank you. Thank you very much. And thank you very much to ISA and Dr. Ola and staff at ISA for the invitation to talk to you today. And I'll be sort of talking about one step back from the detail that Dr. Agala talked about, and I'll look at more at a global perspective, so it's a bit of a step back. Right, now I don't know if those pictures on the side are interfering or not, whether we can remove those. Okay, I'm going to start out with what does the term new breeding techniques or technologies, NBTs, can include? And let's see if I can get rid of those. Yeah, there we go, for mine anyway. NBTs encompass a variety of methods that modify the DNA of plants to introduce desired traits. And so they can include genetically modified plants, GMOs, but with time, the term NBT has become more restricted to techniques of gene or genome editing. And you'll see I prefer the term gene, not genome, because of the precision. Genome implies large scale and gene implies precision. And so gene edited plants may or may not be GMOs, depending on whether introduced DNA is present, and if so, its origin. So presence of unrelated DNA means that a plant is a GMO. No external DNA or introduction of DNA from the same gene pool may or may not be classified as a GMO, depending on the jurisdiction, in other words, the country. So it depends on the circumstances. So as a simple comparison of conventional and new breeding techniques or technologies, in conventional breeding, well, first of all, I would say absolutely that conventional breeding is genetic manipulation, because you're making new combinations of genetic material, and there are crops like triticale, which are completely novel. They don't occur in nature, and there's large chunks of rye DNA in wheat that's grown, but it's not done through recombinant DNA, not classified as GMO, but quite clearly it's genetic manipulation. Anyway, the basis of conventional breeding is, so shall we take, you want to introduce a beneficial gene in blue from a wild relative, you make a cross with the current cultivar, you get an F2 generation, and after back crosses, you will get a new cultivar, which includes the beneficial gene, but of course, you can see that it has unwanted genes. There's red, you've got linkage drag, so it's not very precise. In transgenesis, that gene, the blue beneficial gene, can be isolated. It can be transferred into that current cultivar to give you a new cultivar. So that's much more precise. You don't get the unwanted genes that you would through conventional crossing. And if you look at the terms cisgenesis and intragenesis, then the technology used is exactly the same: gene from related species for cisgenesis, same gene pool, or intragenesis from the same species or even the same individual. Now, initially, transgenesis, cisgenesis, intragenesis were regarded as GMOs and genetically modified. But as time has gone on, new breeding techniques or technologies have, if that genetic information is derived from the same species and could have been introduced by conventional breeding, whether it's cisgenesis, intragenesis, or gene editing, where you can take a current cultivar and make a targeted mutation to get a new cultivar, as we heard in the previous talk. It's becoming, NBTs are now being regarded more as towards gene-edited crops as opposed to GM. So why are these definitions of GMOs and gene-edited produce so important? And the reason for this, it is very important because we have an opportunity to separate the regulation of GMOs and gene-edited produce, because there is 30 years of baggage on the regulation of GMOs. And we have that opportunity now to break that nexus between GMOs and gene editing. And this difference will increasingly enable trade and world trade in gene-edited produce. But before we're going to get that, alignment or harmonization of international regulations is vital, so that producers and consumers can gain the full benefit of these new technologies of gene editing. Now, why do we have to have harmonization or at least alignment? And I think this diagram on the global food system shows how highly complex and interconnected it is, so that you can see that the world food importing and exporting, there's a massive flow of food. So the scale in these is 5 billion, 10 billion, or 20 billion, and there's a different scale of the colors. And you can see from North America, from South America, Europe, Asia Pacific, and Australasia, there's a massive flow of food products from countries which produce grains, for example, and others that produce don't produce grains but produce cereals. So this is another way of showing that. This is non-livestock agriculture and the trade between the top 10 producers, shown in pink on the left, and consumers on the right. And what you can see is that United States, Brazil, Australia, Argentina, Canada are major exporters of food products. And consumers, China is a major consumer. You've got Mexico, Japan, Indonesia, and so on, India. And you can get, of course, some countries are both consumers and producers. So, for example, India is a consumer, but it does actually export some wheat, for example. And United States is a major exporter, but it does also import, you can see, from some other countries, for example, Canada. So there has to be some alignment of regulations in the gene-edited produce before we can gain the benefits of this technology. So there have been rapid advances in the science, as I'm sure you're all aware. So in genomics, the complete genomic sequence of all major crops are now available, and indeed for many of the pests and diseases. And so what that means is the genes that underlie agricultural traits can be identified, and then there are now new ways of combining genes and speeding up breeding processes. So that might be through techniques of conventional breeding, or speed breeding, for example, genetically modified crops, and now gene-edited crops. So I'm going to include a bit about GM, because, for example, if you look at in 2024, more than 90% of maize, cotton, and soybean in the US was GM. So you can see the increase over the last 30 years. And you have to remember that 10% of the world's food is genetically modified, so it's classified as GMOs. And you'll notice I don't use the term genetic engineering, because I'll come to that and why in a moment. So those are GM varieties. Now, these are hybrid herbicide tolerant or insect tolerant BT or combinations for soybean, cotton, and corn. So that's a major, it's been a major success. And this technology, of course, is moving into many other crops. So here's BT brinjal in Bangladesh. And when I looked at this, there were a number of varieties being adopted by, in that case, 27,000 farmers. And those farmers reported a threefold increase in yield at half the production cost of non-BT brinjal. So it shows you, again, this is a Bacillus thuringiensis toxin giving resistance to Lepidopteran pests. In Australia, again, if we look at cotton, in the left-hand side, you can see there's a 99.5% uptake of BT GM cotton. And you see on the left, unsprayed, and on the right is what you get if you do not have 10 to 15 sprays of insecticides. So this is a no-brainer. And similarly, this is Roundup Ready canola growing in Western Australia. And there are two other sorts of herbicide tolerance. They have to be through mutations rather than through GM. And then for improved food for consumers, here's some high anthocyanin purple tomatoes produced in John Innes and sold on sale commercially for in the United States. And they're very pretty, but very also they are beneficial to consumers. I would also like to mention that the one crop that's actually missed out on these GM technologies is wheat. And Bioceres Crop Solution has been developing drought-tolerant wheat and soybean in South America. And they're using the HB4 gene, which is from sunflower. And that will give, under conditions of drought stress, can give 10% increase in yield of soy and a 20% increase in the yield of wheat. So this is something, and there's no yield penalty when there's normal wheat. So this is something which is now Bioceres and their partners are actually developing for the United States as well, Australia, and also countries in the Asia Pacific region. And there's already 50 or 60,000 hectares of HB4 wheat being grown in Argentina and neighboring countries. So this is coming along. This is a company then that had the courage to take this forward. And indeed, we're interested in drought-tolerant wheat in Australia because that's a limitation. And here is some HB4 wheat, or the HB4 gene introgressed into wheat and germplasm in a glass house at Murdoch University. And here's my colleague, John Fossiliaco. So to get an overview of this, these are countries which have approved GM crop cultivation over about the last 30 years. And what you can see, I'm not going to go through all the list, but you can see on the left, all the different crops, the GM crops that have been grown. And you'll see that North and South America are pretty much all of those countries have been growing GM crop, even in Portugal and Spain, in Europe, some countries in Africa, and again, countries within the Asia Pacific region. So, and those in many cases, or most cases, those crops are still being grown. I'm put in a bit of science here because it's sort of evolving. So one area of transgenics initially was RNA or gene silencing, where if you made a double-strand construct which makes hairpin DNA, then as a natural process that is cut up into small interfering RNAs, so 21 to 24 nucleotides, and then you can see that the red strand of that double-stranded RNA, if it recognizes a messenger RNA of the same sequence, an Argonaut protein will cleave that, so it will silence and prevent the expression of that gene. So this is actually potentially powerful technology, and we've used this to develop resistance to root knot, root lesion, and cyst nematodes, for example. So here in this panel, the top histogram, those are controls, and then different transgenic events. And if you look at these are root knot nematodes stained with acid fusion, and you can see the controls of the top three, and the others are different lines in which a silencing signal, so these small RNAs, have been delivered from the plant to the nematode and given a very effective reduction in infestation. But these have to be transgenic. So they deliver that double-stranded RNA, which doesn't do anything to the plant, but it inhibits activities in the nematode. And you can apply that to other organisms, so that, for example, that feed on plants. So this is RNA-based resistance to green peach aphid. And what you can see, again, is wild type or GFP controls, and then very effective resistance, host-induced gene silencing against those aphids. I put that in because those have to be transgenic, but over the last 10 to 15 years, there has been a change and what was limiting application or other sorts of applications was in fact the cost of making double-stranded RNA for agricultural usage. But now that's changed. That's gone down, and spray-on double-stranded RNA, known as SIGs, now is becoming a commercial reality. And that, of course, is not, because you're not using DNA and it's not transgenic, that is not regarded as GMO. So these double-stranded RNA sprayed on is non-GMO in Australia and these other countries here. So that evolution from host-induced gene silencing to spray-induced gene silencing is another change where we also need to look at the regulations. So it's a biopesticide. So getting on to new breeding technologies and gene editing, the most commonly used technology is CRISPR-Cas9, where the development of a single guide RNA, this green piece here, together with a CRISPR, which Cas9, which is a double-stranded nuclease, means that you can target a mutation to a specific site in the DNA, and that then, if you don't get correct repair, non-homologous end joining, you can get insertions or deletions. So that's known as site-directed nuclease 1. If you add a donor DNA, so homologous dependent repair, you can add additional bases at that site, and that's called SDN2. And SDN3, you can add a whole gene, for example. So site-directed nuclease definitions are important. The power of SDN1 is now well known, and I'm not going to give many examples. This is all 6 editing of all 6 alleles of hexaploid wheat to give resistance to powdery mildew. Another one to prevent premature sprouting of grains, and the bottom one, multiplex gene editing of the gliadins, Gamma-Noma gliadins, which are responsible for wheat allergens. And in this work from the Spanish group, they have reduced these allergens in wheat by nearly 98% in one go, which is something you just cannot do by conventional breeding. So these are really powerful technologies for crop improvement. And as was mentioned before, Tropic, which is a company which is based in the UK, is launching its non-browning, gene-edited non-browning bananas. So these are the ones on the right. And they've got another coming, extended shelf life, which is coming in the pipeline. And they have another technology where, in fact, they combine gene editing and RNAi in a form which is not regarded as non-GMO. So this is a new technology coming along. Okay, so in the gene editing kit, there's a lot of advances, right? So SDN1, 2, and 3, DNA-free gene editing, so you can introduce the editing ribonuclear protein combined without any DNA. There's base editing, prime editing. You can have gene segment deletion, gene expression changes, and epigenetic changes, for example. So obviously, I don't have time to go into these. If we compare these, then this is the site-directed nuclease application. I've added another one. So SDN 0, 1, 2, or 3. Is that targeted to a specific genome location? Yes, in each case, as defined by the specificity, nuclease specificity. Does it have a repair template? No, for SDN 0 and 1. Yes, for SDN2 from the species own gene pool. And yes, SDN3, but it could actually include species, some other gene pool as well. And then what sort of changes? Well, SDN 0 is epigenetic changes only, so transcriptional control, but no change to the actual sequence. SDN1, spontaneous mutations, deletions, and substitutions. SDN2, predetermined mutations, in particular, for example, allele replacement, which is what plant breeders try to do all the time. And then SDN3, additional sequence at the targeted location in the genome. So there's the precision coming in. If you can define where DNA is going to be inserted in the genome. So what's in a name? Differentiating GM and GED, this terminology is so important. How you describe it is critical. An imprecise academic terminology, if you're only interested in publications, will be picked up and used by anti-gene technology lobby. So the terms GM, GMO, LMO, GE, and biotech refer, as far as I'm concerned, into introduced genetic material from an unrelated organism. Whereas NBTs, genome editing, gene editing, GED, precision breeding, no genetic material is introduced, or if it is, it's from the same gene pool. So my plea to you is, please do not use the abbreviation GE for gene editing. Use a different one. It's got to be GED or GNED, because if you use GE for gene editing, this simply adds to public confusion, because GE has been used for genetic engineering for 30 years. In fact, my preferred definition is precision breeding, which is used by, has been developed in the UK, but and that is there in their legislation, and they don't use any of these terms. They use precision breeding. So be careful how you describe what sorts of changes are being made. Okay, so in terms of the techniques being used and which countries are using them, well, you can see this is the number of publications. And you can see that CRISPR-Cas is by far and away the greatest technology used. There are other ones: TALENs, base editing, zinc finger nucleases, large oligonucleotides, or prime editing, very much more minor. We look at the countries where the work's being done. China is 2 to 3 times more than any other country in the research going on there, followed by USA, Japan, Korea, and so on and so on. India, Australia are down here, but 41 other countries. So gene editing is now being used very, very widely. So say if we look at those methods used, again, SDN1, 96.6% of publications are using SDN1 gene editing, 2.6% SDN2, and a very small proportion SDN3. And if we look at the species involved, well, rice is the most editing work is being done on rice, followed by tomato, maize, soybean, wheat, potato, tobacco, barley, and 54 other species. So again, very wide application and research R&D going on all crops. Why are we interested in regulations? Well, I come from Perth, down here in the southwest of Western Australia, and Australia is a food exporting nation. For example, 95% of the wheat produced in Western Australia is exported mainly to the Asia Pacific region. And you can see the value of the wheat barley, canola, and horticultural exports. And so, in fact, Australia is the largest exporter of barley and malting barley in the world. So we need to consider what do we need to consider to deliver gene-edited crops? And one is the definitions of SDN2. So SDN1, pretty much people agreed on that, but SDN2, I think we're coming to a general agreement on this, on the definition. Demonstration of absence of external DNA, i.e. from a different source, the possibility of off-target edits, and unintentional low-level presence, which I won't mention. So absence of external DNA, if you have a successful edit, and if, say, you've used a plasmid supplying that editing machinery as tDNA, once you've identified self and selected for null segregants for tDNA, so it only has the edit but doesn't have a construct that would does the editing, because the editing occurs elsewhere from where the insert is. The question is, could there be any residual external DNA from that plasmid? What tests are reasonable? And I think the three main things are sequencing of the site of editing, obviously, quantitative PCR to show absence of backbone of any tDNA, and check sequences at the site of editing to show there are no short open reading frames present. And if there are, to check those against any site, any of the sites to show that there is no allergenic possibility of an allergenic peptide, for example. One thing that people get excited about are off-target edits. And these have got to be viewed in context. Spontaneous mutations occur naturally, 10 to the -7, 10 to the -8 per site per generation. Pangenome sequences, that's of many different individuals of the same species, show many sequence differences in a species. Also, there are well over 3,000 crop varieties generated through chemical or radiation mutagenesis, which actually the difference is caused by double-stranded breaks in the DNA. And there you have large numbers of those, but you can buy those in organic shops, for example. Somoclonal variation, tissue culture-induced variation, is also a major source of mutations. And off-target edits can be minimized by choice of guide RNA, for example. But in particular, there is a history of safe consumption of plant foods with many mutations, regardless of those origins. And an off-target edit, if it resulted in an undesirable phenotype, would normally be eliminated in subsequent crop breeding and testing. So, is there any biological significance in food crops? And I could argue essentially no. If I were doing medical side and you were correcting a defect in an embryo, that's a different story. But in crop plants with this background, this is grossly overstated, this issue. Now, we've been looking with my colleague Mohammed Adil at how to enable trade in gene-edited produce, because of course we were looking to be first movers in applying these NBTs to crop improvement. And in the project we had was to provide information on new science and technologies, policies and regulations that relate to them in Australia and with our trading partners, particularly in the Asia Pacific region, and to help promote international harmonization and alignment of regulations. And also to look at science diplomacy, and how do people come to agreements at an international level, and that's particularly Adil's expertise. So we've run a series of workshops and webinars, particularly with ISA, initially over the as webinars because of COVID, but then, for example, the picture on the bottom right is in Kuala Lumpur, when we could get back to face-to-face meetings. So we've run a series of training workshops, many with ISA, ASCA, APSA, Crop Life, and so on. Here's the ASCA short course in Bangkok. You'll see some people you might recognize in these pictures. There was also a meeting there for the industry side, and I'm very pleased to meet the Director General of the Department of Ag of Thailand, and at that time he announced the deregulation of SDN1 and SDN2 gene-edited plants for Thailand. So that was a great advance. So we've run major meetings in Australia. We ran this one at the Academy of Sciences in Australia. I won't go into the detail, but that covered the science of gene editing, regulations, IP and licensing, industry perspectives, public acceptance, and future trade in gene-edited produce. And that's a lot of some of the background of that with our colleagues in the Asia Pacific is in this publication in Plants, which I refer to as a 19-author publication. And that describes the pathways to deregulation which were available at that time. Here is, for example, in Australia, in fact, it's SDN1 only. So it starts with Institution of Biosafety Committee, then Office of Gene Technology Regulator. So Australia has a very well-run regulatory system. And the questions are asked, is new genetic material introduced? No. Or it's SDN1. Does new breeding method use a transgene temporarily? No. Not subject to regulations a GMO can treat as a conventionally bred crop. Has transgene, for example, a selectable mark being used temporarily? Yes. Is it the final product free of transgene? Again, not regulated. But in Australia, if it's SDN2 or 3, that is still subject to regulations of GMO and can be commercialized as a GMO. So SDN1 only in Australia. And that's because these regulations were looked at probably before most other countries. And so there was a cautious approach. Japan has probably got the simplest and most advanced regulations. And they say, well, have externally processed nucleic acids been introduced? No, not subject to regulations of GMO. Well, there's a voluntary notification. If yes, then subject to GNO regulations. But if that DNA is from the same gene pool, they're not subject to GMO regulations. And there are already commercial products like the high GABA tomatoes and fish on sale, gene-edited produce in Japan. Similarly, India has gone ahead very rapidly. So it's a similar sort of process going through, but I probably haven't got time to go through that, but they have deregulated SDN1 and SDN2. China, in principle, they are advancing, but in fact, gene-edited produce has to go through the same safety evaluation as GMOs. So although they've got all that research and development going on, there's still it is still difficult to get gene-edited produce into the commercialized. In moving further abroad to Europe, in England, for example, the Genetic Technology, sorry, I'm going to go back. Genetic Technology Precision Act 2023 is before Parliament. Well, it's just been passed on the 3rd of March of this month. And the view there that this legislation is crucial for unlocking the potential of precision breeding to develop more nutritious, sustainable, and environmentally friendly crops. And regarding the EU, they say with little progress and stalled editing regulations in the EU, Britain has a golden opportunity to steal a march on the European counterparts and to deliver a vision of making UK science and technology superpower. So this is one advantage UK sees out of Brexit, at least the scientists do, that it's not held back by the complexities of the EU Commission, Council and Parliament, where they have, for example, voted to ban the use of patenting in gene-edited plants. So that's effectively torpedoing incentive for innovators to invest. So this is why you have to get the regulations correct. And so if we look to North and South America, the regulations in place to approve growth of gene-edited plants are in place in North and South America. So United States, Canada, Argentina, Brazil, Chile, Paraguay, Uruguay, Colombia, Guatemala, Argentina was one of the leaders here. And in Africa, Nigeria, Ghana, Kenya, amongst the leaders, Malawi, Uganda, Zimbabwe, Ethiopia. There are regulations moving forward in Mozambique and Botswana. South Africa sort of made an advance. It seems to have gone back, but anyway, we'll see what happens there. So this is my update of global regulations, global status of gene-edited regulations. So red is prohibited or restricted, green is approved or approved on a case-by-case basis. And so you can see only a few countries in the Americas which are prohibitive. Yellow is basically an ongoing debate with approval likely, so including countries like Cuba. You can see that the UK and particularly England is going ahead. Albania is a little red country in here. The grey is some regulatory progress. So Europe's made some progress, but it's still a bit behind. And you can see the Francophone countries are unfortunately following Europe, whereas the ones that have been part of the British Commonwealth, on British-speaking ones like Kenya and Uganda and the eastern side are progressing rapidly. And again, there's been rapid progress in the Asia-Pacific region. The one country that's not progressing so far is Malaysia, which is a shame. And I should be changing New Zealand to yellow because now there is now progress towards approval of gene-edited produce, and that's coming along. So what you can see is that the whole area of the status of regulations is changing very rapidly very positively. So when will it be possible to trade in gene-edited produce? Well, North America, USA and Canada now. South America, so these Latin countries have their own gene editing trade agreement. The EU, well, I've covered that. The problem there, it may be there, well, the revised proposals will be passed in 2026. We'll wait and see. In the UK or in England, you can get gene-edited produce in the field in 20 days. So that is a fantastic advance. In the Asia-Pacific region, trade in gene-edited produce is now starting. It's certainly possible between Japan, Philippines, and Thailand, particularly, for example, with that high GABA tomato. And African countries are advancing, particularly Nigeria, Ghana, Kenya, and the others coming along. So actually, it's a very exciting time. The regulations are advancing rapidly. They're pretty much all going positively to accepting commercialization of gene-edited produce and aligning, if not harmonizing, regulations. So we now have the opportunity really to reap the benefits of this technology, as long as we have science-based policies in each of these countries, that they don't only consider internal regulations, but we have to look at the international trade part of that. But otherwise, it's exciting times in this field. So I'll just thank you. I hope I haven't gone on for too long. And I will end there. Thank you. So I'll stop sharing.
Thank you very much, Dr. Jones, for that very comprehensive presentation. Definitely the presentation of our speakers has given us more knowledge about the global and local perspectives on the regulatory frameworks for new breeding techniques in crops. Again, thank you very much, Dr. Jones. Everyone, let us give him a virtual round of applause. So that concludes our presentation for today. We encourage everyone to post your questions through the Zoom Q&A function. For our Q&A session, we have the Senior Associate Scientist of ISA, Mr. Panfilo de Guzman, to moderate the discussion. Sir Poppen, the floor is now yours.
Okay, thank you, Janine. We have quite a lot of questions in the Q&A box, and some of it was already answered by Malu. But I think I have to, for the benefits of the other participants, I may have to read again the questions answered by Malu. and perhaps ask her to if she can add more to her previous answer or if she can expound more. So there is one question about how is the efficacy and durability of resistance to diseases ensured for gene-edited crop that is not covered under the GM regulations? And maybe Dr. Jones can also add to that. Your answer, can you add something about that question, Malu?
Um, yes, aside from, well, when I
Mm.
say that since this gene editing techniques are really very precise and are really very targeted, so it's really uh, it really allows our scientists to make these targeted changes. Like you can actually indicate where in the genome where in the genome we will target that particular trait, and then you can actually make targeted or make targeted changes in that particular genome. So this is really very precise. So you can ensure that um, the trait for resistance or durability for resistance is being actually like you can delete or you can insert or you can like make some few base changes in the genome of the particular crop.
Mhm. Okay. Can you add anything to that, Dr. Jones?
Now, I think if I look at the Q&A on that, was that referring to GM crops in particular rather than GM? It looks like it might be, so I would say first of all that if you take, for example, GM cotton, to be honest, that's a no-brainer. If you have genetic resistance to cotton bollworm, for example, you instead of having to spray your crop 10 to 15 times to control insect pests, you may spray once because there are some other pests. But what it means is that genetic resistance gives you much better result and much fewer off-target effects on other insects and beneficial insects than conventional control. So that's an absolute no-brainer, and it is so beneficial. And that is why all the world's major cotton growing areas are using BT cotton, for example. And so we're talking now 30 years experience on this. In terms of gene editing, well, what you have to do in a way, when I talked about RNAi, and there you can deliver silencing signals to a pest or disease. So not only nematodes and aphids, but you can deliver those to fungal pathogens and viruses as well, actually. But they have to be GM. If you want to use gene editing procedures, then you probably have to turn everything around and target genes to susceptibility. So, for example, that MLO gene is a gene susceptibility to powdery mildew in wheat. And so one base change there through gene editing can give you resistance. So if all the copies of the MLO gene have one base change in a particular site, and then you have resistance. So this is very targeted and very specific, and has no effect on off-target effects on other organisms. So this is the precision of gene editing is very what's one of the gives us its power and specificity.
Okay. Since the last few slides you presented, Dr. Jones, is about trade of genetic products, my question is, what could be the possible challenging trade scenarios with the asymmetric regulation on gene editing among different countries? So they have different regulations, and what could be the possible trade scenario with that kind of differences in regulation?
Sorry, I seem to have gone back into my slideshow. Let's see what's happened to it. Zoom. Where have I gone? Post Zoom. I don't know what I've clicked on. Well, I don't know if you can see me.
Yeah. Mhm.
Can you just repeat that question, because I seem to have lost my Zoom
The
picture.
question is about possible trade challenges
Ah,
when it comes to having asymmetric regulations among
yeah.
countries who are trading the gene-edited products.
Well, it's just like GMOs. If a country which is importing produce has not got its regulations in place, then the same sort of issues as for GM produce would occur. In other words, it could get rejected at a port of entry. So that's why it's so important for anybody that's involved, for grain handlers, exporters, marketers, and exporters have to know what the gene editing regulations are in importing countries, and to make sure that their products are compliant with that country. So that's an absolute requirement. That's why you had to have alignment of the regulations in all of these countries before we're going to get large-scale trade in gene-edited produce. And that's why it's so important that those countries which import food need to get their regulations sorted out internally, but they should be internationally consistent and not just based on internal considerations. Because if they're different from other countries, then again, it makes life very, very complicated. And I think it's moving that direction. I'm very optimistic.
There is a question for you, also related to trade. It's from Russell Rank. So he says, I understand the separation of GMO and gene ed on the basis of the extent of DNA introduced source of DNA. Since regulatory is focused on safety, it implies that there is a safety difference, in risk between these methods, but to my knowledge, there is no difference in risk. Can you comment on this? Are there risk differences of which I am not aware? So I think he's talking about this are more of a technical and the science between between GM and gene and technology. Are there any differences in risk?
Well, we have had 30 years of GM crops, and at the start of it, people raised all these risks about super weeds, and other potential risks. And we know after 30 years of experience, in fact, and 100 large-scale examination of potential risks, including in the EU, that spent half a billion dollars on this, and they found that the risks are no different from conventional breeding. So let's get it in perspective. In conventional breeding, there are risks. Because you don't know, for example, what other genes you're transferring when you make crosses, and people don't bother to look. There are 3,000 products of irradiation breeding on the market. Why nobody bothers to look, and yet they have large numbers of double-stranded breaks, which all over the place, where nobody knows where they are, but they haven't used recombinant DNA. They're imprecise, but you can grow and sell them. Whereas it's the wrong way around, because if you're going to have regulation, those products should be more regulated than gene editing, where you have very precise changes at precise sites. And so I think the risk from gene-edited produce is much lower than conventional breeding, and certainly much less than, for example, mutagenesis breeding, which is still widely practiced.
Okay. There's a question here from Malu, and I think she answered it already. If Philippine regulation is product-based or process-based, and I think it is our regulation is product-based, right, Malu?
For
So
NBT, yeah,
for
we
NBT,
actually, yes.
okay. For gene-edited, it is product-based.
Like it's a combination of the technology and the product.
Okay. Dr. Jones, for the other countries that you presented, which countries are product-based or process-based when it comes to gene editing technology?
Well, Canada, for example, is product-based. Most countries are process-based. So, for example, Australia, if at some point you use recombinant DNA technology in the process, then that triggers the need for regulations through the Office of Gene Technology Regulator. If then you provide information that there is no external DNA or from the same, or in the case it's no external DNA, then that is no longer regulated. But actually, product-based would be, on the whole, would be better because that is what a conventional breeding is based on. Now, we haven't really touched on foods, on the food side of these and acceptance. So in addition to Office of Gene Technology Regulator, in Australasia, there's Food Standards Australia, New Zealand. So they're concerned with food safety once it's food. So gene technology itself, that's Office of Gene Technology Regulator, and for food, then that's Food Standards Australia, New Zealand. So there are different regulations as to whether you're dealing with a living organism or whether you're dealing with a food product. But in most cases, in Australia, we're still waiting for FSANZ, Food Standards Australia, New Zealand, to come up with their latest regulations. But I'm not sure if I've gone off the topic here because, as I say, I seem to have lost my picture on this bit. Does that answer the question at all?
Yeah.
Oh, yeah, so it's
Yeah.
product versus process.
Yeah.
Yeah, so mainly it's process, but in countries like Canada, it's product.
There is a question here from Saarani, Bangladesh. For you, Dr. Jones, in your opinion, to regulate NBT, countries should revise their current GM framework or establish a new guideline for NBT?
Well, that's a good question. It depends on what the framework is for GMOs and whether it can be adapted. So in Australia, it's one. The Office of Gene Technology Regulator is based in the Department of Health, and it covers health, industry, animals, plants, new vaccines, for example. So that makes it very slow to respond. Whereas I've talked to another country I've dealt with, Mauritius, where I said, well, look, you probably, if you can make your regulations for gene editing within the Department of Agriculture, or that's current name, then you have the opportunity to change the regulations and be as technology advances, you can modify. If you only have it in one monolithic structure, then making changes is very difficult. So it depends on the background. If you were starting from scratch, it would be it would be good, I would think, to separate, for example, the medical side from the agricultural side, because these are very different things with very different needs and requirements and speed and so on. So yeah, if you're starting from scratch, you might just, for example, from the crop side, keep that as a separate set of regulations within the Department of Agriculture and associated organizations, and then it makes it much more flexible to change things as technology advances, which it is doing very rapidly.
There's one question here for Malu. For SDN3, it combining different genetic elements, although coming from the same species, and editing same crop species, for example, rice promoter, gene and termination sequence, is it considered non-GM in Philippine regulation? I think the participant is referring to SDN3.
SDN3.
Yeah.
Okay, well, actually, what I said earlier is that we have a very simple regulation on NBT. Like, if the product contains a foreign DNA, then it will be considered a GM, and if not, then it's non-GM. So, I think we follow that simple policy on NBT.
Okay. Thank you, Malu. question for Dr. Jones. Do you have any insights about why we are not seeing more commercialization of gene-edited crops yet, despite many countries no longer regulating them as GMOs?
Well, anything to do with agriculture takes time. Any new technology has to
Right.
go through the system. So, it's not something completely separate. It is based on, for example, in crops, you will take the best genotypes for that particular crop, and then you have to make sure that you can do the editing on that. So there's a tissue culture procedure, there's making, so it takes time to go through all the processes. But it is definitely clear that to get gene edited produce out is now much quicker. So in the US, for example, non-browning mushrooms and other produce has come through very quickly. In Japan, the tomatoes, the fish, and other produce are already there on the market. So, but you cannot edit, at least on a large scale, say a crop like wheat in Australia, where we export. Western Australia, we export 96%. We cannot convert that to gene editing until we know the countries. For example, like Indonesia is #1 importing country. They've got to have their regulations in place and aligned with the Australian regulations before we can export to them. So the alignment of regulations internationally is the key to greater application of gene editing technology. If it's just within a country and there's no exports and there's not a possibility that material will end up in some produce which is going to be exported to a country that doesn't have its regulations in place, say like Malaysia, then internally it can be used. And for example, in Argentina, that's the case on the Latin American countries. But I think it's just early days in the technology. The first GM work was done in the 1980s, but the first commercialization occurred around about 95 to 96. So there's always a lag between a technology being from the science of that to be commercialized and become a reality and trade in it. So it's coming along, but there are obvious reasons as to why that takes time. It doesn't happen instantaneously. So you've got your microphone off.
Sorry about that. Okay. So another question for you, Dr. Jones, on the deregulation of SDN1 in Australia. Does it apply to food, feed, and crops?
Yeah, that's SDN1 is for all plants, food, feed, crops.
Okay, thank you very much. Malu, a question for you. What kind of international collaboration is the Philippines pursuing in terms of policy development or capacitating regulators on gene editing regulation and some initiatives on gene editing, developing gene edited products?
Okay. Well, right now we are in constant communication with the USDA because of they want to help us or to collaborate with us on the regulatory framework for GM animals. And also one time I have this just a small conversation with the biotech in US, and they're quite interested in helping us in developing our policy on microbials, because we have this plan of improving our guidelines for contained use of microbials. So we have this initial discussion with the USDA.
Mhm. In terms of capacitating the regulators,
Capacity.
yeah,
Yeah,
have you done any need assessment when it comes to what really the regulators in the Philippines really needs?
yes. Actually, in terms of like in gene editing for animals, and also on GM
Yeah.
animals, we are actually also reaching out in our partners in abroad, if they could give trainings or workshops on
Yeah.
GM animals or on gene editing. And they are not promising anything, but they would always say that they would look into it and would try to help if in any way they can. Yes.
Yeah.
Our
Inci-
regulators really need capacity building
Okay.
for these two technologies.
Incidentally, our next webinar will be on gene editing regulation for animals. So that will
Oh,
be
that's
maybe
good.
2 weeks from now. A question for you, Dr. Jones. For regulators to be certain that external DNA is absent, is it necessary to require the product developer to provide PCR of the whole tDNA regions? This is a more technical question, I think.
Yes, I mean, I think the three things which I said were the minimum, which is you do PCR across the you sequence across the edited region. You do a PCR which will cover all sequences which would have been in the construct that if a construct was used to introduce the guide RNA and the Cas9 to show that that is absent. And if there are changed sequences at the site of editing, whether any new open reading frames or small peptides could be produced, and if they were, whether you could compare those with the peptide databases and see whether there's a possibility or any suggestion that there might be some new allergenic peptide produced. I do not think it's necessary to do total genome sequencing. If you talk to people who run sequencing laboratories, they say, oh yes, because of course they want the work. But I think it's totally unnecessary. For example, the genome of wheat, which you have in the background of this picture, is 5 times a human genome, 17 gigabases. It then adds to adds to the cost and slows down the whole process. And yet, when you have changes like this for example, in a result of chemical radiation mutagenesis, there's no test whatsoever. You don't even have to care about any of those things. So I think we must be careful about not overburdening developers with regulations which are totally unnecessary.
Okay, I think we have covered most of the questions in the Q&A box. But before we proceed with the closing of this webinar, I would like to know your insights, the two of you, Dr. Jones and Malu, on what could be the future of gene-edited crops in the coming decade due to the different regulatory processes among countries producing gene-edited products. Dr. Jones, you please first.
Oh, well, I mean, I think, as I said, I've been maintaining sort of world maps of the progress of the regulations, and I keep on having to update them. So, for example, I should have updated at least New Zealand to yellow, because that is actually, there is change there. EU, I hope within a year or two, we can actually, they will actually get their act together. So I think that the regulations or people are realizing that there's limited amount of land in the world for food production. In fact, there's becoming less per person. We've got to maintain the natural environment and biodiversity. We have to maximize the production on currently cropped lands and not go to the wilder areas where biodiversity resides. So the only answer is to increase production. And although gene editing is only one opportunity, because there are improvements through the new sequencing technologies and haplotypes. There's speed breeding, there's much faster marker-assisted breeding. So it is one part of the whole picture of increasing productivity. But I think it's a very important one because it's so powerful in what can be done, how precise it can be done, and how rapidly it can be done. So we can now reach the same end product of conventional breeding, but much more accurately and much faster. So I think that is being recognized, and I hope, and I do believe, say, if you say 10 years time, I think that the majority of countries in the maps I showed you will have gone to green. And then that enables large-scale trade in gene-edited produce. And again, we will have experience of the fact that there, and I'm sure it'll be the case, that there's no untoward effects, or certainly no more risks, and probably less risks from conventional breeding.
Okay. Malu, what would be the importance of, to your mind, the regulatory alignment within the region?
Yeah. I think in terms of regulation, I'm aware that there are harmonization efforts through organizations like the APEC, OECD, and Codex, but that could help in harmonizing all this regulation. And as I can see that there are many countries, or more countries, are developing clear guidelines on how to regulate or distinguish gene editing from transgenic crops. And there are actually international discussions, ongoing discussions to align standards and promote the mutual recognition of safety assessment of these gene edited crops. So I'm more positive. I'm positive that in the future we'll have this regulation which would really help in trading these products.
I mean,
Okay.
could I add,
Yes,
add
please.
one thing here? You know, at the international level, that's where a lot of what holds up the harmonization and regulation. So we have a series of MOPs,
Yes.
meetings, and so on. And what you have there often are regulations or people meeting who are don't understand the science, and so and also that they are given from non-scientists. For example, it might be in the Department of Foreign Affairs or something, who are non-scientists, provide the paperwork to say this is our position without really understanding it. And so the science diplomacy I mentioned, which my colleague Adil has been involved with, and he's now senior secretary in the Pakistani embassy in Brussels, and he's one of the things that he is pushing hard is to try and get science-based scientists and science-based decisions coming out of these international meetings, and not based on politics and not based on interest groups, which unfortunately don't have, I think, our best interests of the consumers, producers and consumers at heart. So that's one thing which is a little bit of caution that we need to be aware of as well.
Okay. With that, thank you very much. I think we need to give our two resource persons virtual round of applause. Thank you very much, and I give back to you, Janine, the floor.
Thank you very much, Sir Poppen, for moderating the Q&A session. As we move forward, we encourage everyone to please complete the evaluation form to receive your certificate of participation. Scan the QR code flashed on your screens or go to the link to access the evaluation form. This post-webinar survey will help us improve our future events and webinars. Again, you need to complete the evaluation form to receive your certificates via email. For everyone's information, we will be having the second part of this webinar series focused on animal biotech regulations. If you're interested in joining the said webinar, please do follow us on our socials at isa.org or subscribe to ISA's weekly newsletter, Biotech Updates. Stay tuned for our future announcements, and we hope to see you there. Before we wrap up our today's webinar, we would like to extend our sincere gratitude and appreciation to our resource speakers, Ms. Malu and Dr. Jones, who shared their expertise on regulations. Your insights and knowledge are invaluable, and we truly appreciate the time and effort you dedicated to making this webinar a success. We are truly honored to have you as our speakers, and we look forward to future opportunities to collaborate. To formally close our webinar, let us watch the recorded video message from the Director of DA Biotech Program Office, Dr. Paul Limson.
Good afternoon, biotech enthusiasts. Congratulations to everyone who participated in today's webinar, and a heartfelt thanks to ISA for organizing this invaluable opportunity for knowledge exchange. In recent years, we have witnessed significant advancements in biotechnology, particularly in the realm of new breeding techniques or NBTs. which hold great promise for transforming global agriculture. These technologies have the potential to address some of the most pressing issues faced by our agricultural systems through the precise, efficient, and cost-effective development of high-yielding, disease-resistant, and climate-resilient crops. Throughout today's discussion, we've explored various regulatory frameworks from around the world, from the presentations by Ms. Maria Lorelai Agbagala to Dr. Michael Jones. We have learned about the diverse approaches being adopted to regulate NBTs. It is evident that while some regions emphasize stringent regulatory measures, others advocate for a more flexible framework that promotes innovation while ensuring safety. All of these frameworks are critical in ensuring that products are developed and utilized responsibly, taking into account both the opportunities and challenges they present. It is clear that while there is shared enthusiasm for the potential of these technologies, there are also diverse perspectives and approaches to regulation. If there's one thing I want to stress is the importance of building effective communication with the public regarding NBTs. Building trust through transparency and education is crucial for public acceptance and the successful adoption of these technologies. It is our shared responsibility to ensure that stakeholders, from farmers to consumers, are well informed about the safety, efficiency, and advantages of all biotech products. As we conclude today's session, I encourage each of you to continue the conversations initiated here, whether through research, policymaking, or community engagement. The future of agriculture really depends on our collective efforts. Once again, thank you to our speakers for their expertise, our organizing team for their hard work, and all of you for making this webinar a success. Let's continue to engage and work together towards a progressive and sustainable agricultural sector. Thank you and mabuhay tayong lahat.
Once again, thank you very much for joining us today. We hope you learned a lot from the presentation of our speakers. Thank you, everyone.
Thank you, Dr. Mike, and thank you, Malu. Thank you, everybody. Thank you.
You.
Good job, Janine. Good job, everyone.
Thank you po.